This is Chapter 1, The Microbial World and You. Normal intestinal bacteria is part of our normal flora and it helps keep you happy and healthy. Learning objectives list several ways in which microbes affect our lives. Define microbiome, normal microbiota, and transient microbiota. So, microbiology, it's going to be talking about microorganisms that are too small to be seen with the naked eye. Microbes include bacteria, fungi, protozoan, which are single-celled eukaryotes, microscopic algae, viruses, which are too small to be seen with a light microscope, and prions, which are misfolded proteins. Stands for protein infectious. Misfolded proteins that can cause brain diseases are called prions. So microbes, luckily only a few are actually pathogenic, meaning disease producing. As you know, even if you put food in the fridge, it eventually gets rotten, and that is because of microbes. All right, they're part of the food chain in aquatic environments. They decompose organic waste. Here, the word organic means it contains carbon and hydrogen atoms. And they also incorporate nitrogen gas in the air into organic compounds such as amino acids. And they also generate oxygen by photosynthesis. Microbes produce chemical products such as ethanol, acetone, and vitamins. They also do good stuff like produce fermented foods, vinegar, cheese, yogurt, alcoholic beverages, and bread. And they're used in manufacturing such as cellulose and disease treatment, insulin. So pretty much anything you can get a gene for, you can take that gene, put it into a microbe, and produce that gene product. All right, so destructive and beneficial actions of microbes. So microbes are part of our normal flora. That's good because they help compete against other pathogenic microbes. So the adult human has a microbiome, which is about 40 trillion bacteria living in and on your body. So we call that the normal microbiota that lives stably in and on the human body. Pretty much anything that is open to the environment is going to be covered with bacteria. So again, luckily it can help keep the pathogenic microbes away and also trains the immune system. So again, normal microbiota, it actually begins to be acquired before birth, uh, can colonize the body indefinitely or fleetingly, which is termed transient microbiota. So this is the one that you want to know the definition. Transient microbiota, they're kind of just passing through. So colonization is at body sites that provide nutrients and the right environment for the microbes to flourish, right? For example, E. coli is going to live in your intestines, Escherichia coli, because the pH is right and the environment is good for E. coli to live in your intestines. All right, so from about 2007 to 2016, and even going on to today, we've been studying the makeup of the typical microbiota of various areas of the body and understanding changes in the microbiota and how that affects human disease. All right, so now we've begun the National Microbiome Initiative, which began in 2016, that explores the roles microbes play in different ecosystems on the Earth. Okay. So, what percentage of all the cells in the human body are bacterial? A large amount. All right, so we want to know genus and a specific epithet, the name of the species. So, genus and species. Major characteristics of each group of microorganisms and the three domains. So this is one of our early scientists, Carolus Linnaeus. He established the genus and species. So genus will be capitalized, genus, and then species will be lowercase. If you're writing them by hand, you're going to want to underline so everybody knows you're talking about the scientific name, genus, and species. And if you're typing it, you're going to want to italicize it so you can communicate you're talking about the scientific name, genus, and species. Binomial nomenclature. Okay. And they could be named after what the disease does, or some scientists are just greedy and want to name it after themselves. So, for example, Escherichia coli is kind of a two-part thing because Theodore Escherich, he named the genus after himself, and then the species gives a hint that it lives in your colon. So coli refers to colon, and that's your species. Escherichia refers to the scientist who discovered it. All right, then we have Staphylococcus aureus. Okay, so staff means kind of grape-like clusters. Let me turn to purple. So grape-like clusters means staph. All right, and then coccus means round. Okay, so the clusters kind of looks like a thing of grapes, and then coccus means the round morphology of the bacteria, and then it's called aureus because that is gold. If you look on your periodic table, you're going to see the abbreviation for gold is AU from the Latin. So when you look at the colonies on a petri dish, they're going to be gold colored. All right, so again, scientific names, genus, and species, or you can just write the first letter of the genus followed by the species. All right, so genus is going to be a broader group that's going to encompass perhaps several species. Okay, so we want to definitely know all the different types of microorganisms that can make you sick. We have bacteria, archaea, fungi, protozoan, algae, viruses, and then multicellular animal parasites, such as worms. So the word for worms is helminths. Helminths. I think of a little worm wearing a helmet. That's how I remember it. Here's my little guy. This is a helmet. I know you love my artwork. All right, he's a worm and he has a helmet on because here's his little strap. There you go. He's a worm. Hi, I'm a helminth wearing a helmet, a multicellular animal parasite. All right, so there are a bunch of different types of microorganisms. Bacteria. They are in the prokaryotic domain. All right, they're single-celled, in other words, unicellular. They contain a molecule called peptidoglycan in their cell walls. They're going to divide by binary fission. Okay, binary fission, peptidoglycan, prokaryote, bacteria. Important words to know. Okay, and they get their nutrition from organic or inorganic chemicals, or there are some photosynthetic bacteria. And they can also do locomotion by moving appendages called flagella. All right, so flagella, you might have seen them on a sperm. However, bacterial flagella are going to move differently. They are going to move kind of like a boat motor around in a circle. All right, then we have archaea, also prokaryotes, because they have no nucleus. They have no nucleus. So that makes them a prokaryote. They're thought to be older, right? About 4 million years older. Okay, they do not have peptidoglycan, and they might not even have a cell wall. Archaea are going to live in extreme environments, so methanogens, meaning they produce ch04, methane. They are extreme halophiles, meaning they can live in very salty condition. Halophile means salt-loving. Thermophile means heat. So halophiles love salt. Thermophiles love heat. So luckily, we don't have to worry too much about archaea being pathogens, because usually they're not. Okay, our next group is going to be our kingdom fungi. Now, these are eukaryotes, okay? They do have a nucleus. So we are more closely related to fungus than we are to bacteria. Okay, they have chitin or chitin in their cell walls. They absorb organic chemicals. Again, here, organic does not mean you got it from Whole Foods. It means that the molecule contains both carbon and hydrogen. Yeast are going to be unicellular. Molds and mushrooms are multicellular. All right, and molds are going to consist of masses of mycelia that are composed of filaments called hyphae. So if you've seen mold growing on your food, it looks kind of fuzzy. That's because of the hyphae filaments. Okay, so here's what they look like. And then inside they have little sporangia. Then we have protozoa. Again, these are going to be eukaryote. Eukaryote means it does have a nucleus. Okay, we are humans, we are eukaryotes. We have a nucleus, and protozoans have a nucleus inside their cell where they store their DNA. Okay, so these guys can be motile. They can move around using pseudopods, cilia, or flagella. So cilia would be little short, they're not hairs, but they kind of resemble hairs. All right, so cilia, they're going to be the same type of cilia that we have in our respiratory passageways, in our eustachian tube. That is going to move fluid, but instead of moving phlegm and such, these guys are going to move themselves through the water with their cilia. Okay, they can be free-living or parasitic. Some are photosynthetic. They can reproduce sexually or asexually. Okay, so these are the little pseudopod. Pseudopod means fake feet. Fake feet are pseudopods, and that is how these little guys are going to move. You may have learned about white blood cells already, so you know phagocytosis. This is going to go around and gobble up this food particle. Okay, then we have algae. So eukaryotes, again, a eukaryote means that it does have a true nucleus. Eukaryotes have nuclei, so algae are eukaryotes, meaning that they do have a nucleus. Okay, they have cellulose in their cell walls. Cellulose. Anytime you see something ending in OSE, that's likely to mean it's a carbohydrate. So algae can live in freshwater, saltwater, and soil. They're going to use photosynthesis for energy, and this is great because as humans, we need their byproducts. So they produce oxygen and carbohydrates. They can reproduce by sexual and asexual means. Okay, here's a little picture of algae. All right, now viruses, are they alive? No. Nope. Viruses are not alive. Why are they not alive? For one thing, they don't even have cells. They're acellular. Okay, they're going to have either DNA or RNA. Generally, not both. So obviously they can't make proteins. They can't do anything themselves. Okay, so they have a core that is surrounded by a protein coat, and then that coat may be enclosed in a lipid envelope. They can only replicate in a living host cell. So outside, thankfully, they're not reproducing on their own unless they're in a living host. So that's kind of a good thing when it comes to pathogenic viruses. Okay, so this is a virus. Then we have multicellular animal parasites. So eukaryotes, multicellular. Technically, they are not microscopic because you can see them, all right? But they are microscopic in their life cycles. Otherwise, most sane people would not eat them. Now, there have been some people who are crazy, in my humble opinion, that have deliberately eaten tapeworms in order to lose weight. Ew, no thank you. They can cause intestinal blockages, so that's not a great idea. Okay, so we have prokaryotes. Prokaryotes are going to include your bacteria, and archaea. All right, you are a eukaryote. Just think you are a eukaryote. Archaea. Okay, so bacteria and archaea, those are going to be prokaryotes. Everything else is going to be a eukaryote except for viruses are neither one. Viruses are not prokaryotes and they are not eukaryotes. They're not alive. All right, so we have another scientist, Carl Woese. In 1978, he made the 3-domain system. They used to be 5, so be happy. Now we have just 3, bacteria, archaea, and eukarya. Okay, and then within the domains, we have kingdoms. So we have kingdom for protists, fungi, plants, and animals. What are the 3 domains? Remember, bacteria, and archaea. Those are going to be your prokaryotes. Why are they called prokaryotes? No nucleus. They do not have a nucleus. All right, and then we have eukarya. Eukarya, they do have a nucleus. You are a eukaryote, you do have a nucleus. All right, so more scientists. You definitely want to know all these fabulous scientists that we're standing on the shoulders of. So we have Hooke and Anthony van Leeuwenhoek. Okay, they came up with ideas of where things came from. They had a crazy idea called spontaneous generation, that things just pop popped out of the air. Have you ever seen a baby just pop out of the air? No. It has to be created by biogenesis. All right, and then we had these scientists who did experiments and came up with conclusions. So we have Needham, Spallanzani, Virchow, and Pasteur. So, observations. That's our first step in the scientific method. We have Robert Hooke. He saw that there were little boxes and he called them cells. So, the cell theory is all living things are composed of cells. So, the first microscope wasn't really until about 1773 or 1723 and created by Anton van Leeuwenhoek. He said there were little animalcules, so little animals. He looked at bacteria and protozoa in a magnifying lens. He looked at things like vinegar, for example, found little bacterias in there. Okay, so this is how the first microscope looked. All right, what is cell theory? Things are made of cells. Living things are made of cells. So is a virus alive? No, it doesn't have cells. So they used to think that things just popped into the air when there was a vital force present. Our current theory is that of biogenesis, that living cells arise from pre-existing cells. Okay, so Francisco Redi put decaying meat in jars. All right, he covered some of the jars with a fine net, no maggots. He left some open, they got maggots. If the jars were sealed, they got no maggots. Where did the maggots come from? Well, maggots are fly larvae. So a little fly would come along, lay an egg, and then maggots or eggs, maggots would be the eggs generating, biogenerating maggots, which then grow into more flies. Okay, what was the purpose of the seal jars? Okay, that's going to be your control. No maggots. So, yeah. Here, John Needham put boiled nutrient broth into covered flask. Okay, so he put them into the covered flask. Oh no, he did find microbial growth. What happened? Okay, so this guy, Spallanzani, he boiled the nutrient solutions, put it in a sealed flask and heated it, basically killed any microbes, and then they found no microbial growth. So that would support biogenesis. Okay, so then in 1858, Rudolf Virchow stated that living cells arise from pre-existing cells. I think we already, yeah, they didn't know that at the time, which is pretty wild. Seems like common sense now, but they didn't know it then. Okay, Louis Pasteur demonstrated that microorganisms are present in the air. He actually was helping out. The French wine industry had some trouble where the wine was going bad. So he was able to prove that the microbes came from the air, right? He put nutrient broth in a flask, heated, did not seal it. It had microbial growth. He sealed it immediately. No microbial growth. So that is why we call what we do to our juice and things like that pasteurization, right? We heat it up really hot, real quick, and then immediately seal it. And now it's called pasteurized because it doesn't have microbes too many microbes. All right, he did another, Pasteur did what we call a swan neck flask experiment. Okay, so in this, uh, there was no signs of life. So it's kind of like the pew trap under your sink, right? So air could get to it because they said there was some kind of vital force in the air they didn't really understand, and then it would trap the microbes. Okay, so this is what it looks like. Here we have the swan neck flask that's shaped like an S. So just like the thing, if you drop your ring in the sink, it's going to end up getting caught right there. Okay, so the microbes were getting caught down here so that they didn't make their way into the solution, but air was still getting to the flask. Okay, so what evidence supported spontaneous generation and how was spontaneous generation disproved? Okay, so Louis Pasteur did the best one because it was still open to the air. And then Needham made a mistake with his experiment, and it was actually contaminated. Back in the old days, they would transport glassware in hay to keep it from breaking, and his lab was contaminated by particles on the hay. So that is why he got incorrect data. Okay, so Louis Pasteur influenced Lister and Koch. German. All right, so Koch's postulates, you really want to know. I'm going to ask you to list them on your test, okay? There's 4 of Koch's postulates. We're going to talk about Jenner, Ehrlich, and Fleming. Okay, so the first golden age of microbiology, 1857 to 1914. Discoveries and developments included the following: the relationship between microbes and disease, the role of immunity in preventing disease, chemical activities of microorganisms, microscopy got better, we learned how to culture microorganisms, vaccines, very important, aseptic technique, and chemotherapeutic drugs. Now, when you hear chemotherapy these days, you tend to think of anti-cancer drugs, but chemotherapeutics means any kind of chemical that is used to make you better. All right, so fermentation. Pasteur showed that microbes are responsible for fermentation. Fermentation is a microbial conversion of sugar to alcohol in the absence of air. Microbial growth is responsible for spoiled food and beverages, and then bacteria that use air spoil the wine by turning it to vinegar, which is also known as acetic acid. All right, so Pasteur got the idea, and we now call it pasteurization after him, that you could kill the spoilage bacteria by heat, not hot enough to evaporate the alcohol in the wine. So pasteurization is the application of a high heat for a short time to kill harmful bacteria in beverages. All right, so this is a little timeline. Pasteur got the idea for fermentation, disproved spontaneous generation, pasteurization. Lister, you've probably heard of Listerine because he got the idea of using solutions to kill germs with for aseptic surgical technique. Okay, Robert Koch, germ theory of disease. So you do want to know his 4 postulates. So we're going to be asking you to list Robert Koch's postulates, how you determine what microbe is causing what disease? Okay, then we have Nesser. He is the one who discovered Neisseria gonorrhea. Okay, Koch discovered how to do pure cultures, and then Finley learned about yellow fever. Okay, so a lot more scientists. Agostino Bassi showed that a silkworm disease was caused by a fungus. Louis Pasteur showed another silkworm disease that was caused by a protozoan. Remember, that's a single-celled eukaryote. Now, this guy was very brave, and unfortunately, he ended up in the poorhouse for standing up for what he believed in. But we got to be very grateful to him because he is the one who got the idea of washing your hands, right? So back in the day, the surgeons would walk around with bloody coats on and not wash their hands. They would do a surgery on a rotten limb and cut it off, and then they would go deliver a baby. Okay, so that's really gross. They would go from like an autopsy to go deliver a baby. All right, and that's where we got puerperal fever. Okay, so they would spread it from one patient to another, and that mother would end up dying because of the dirty, contaminated doctors who did not wash their hands. All right, so we have Pasteur. Again, he showed microbes are in the air, can spoil food, cause animal diseases. Okay, Joseph Lister, just think of Listerine, right? Easy to remember. He used a chemical antiseptic, and one of the original ones was phenol to prevent surgical wound infection. So he got the idea of cleaning the site with a chemical antiseptic, and it worked. Okay, then we have Robert Koch came up with the idea that bacteria causes anthrax and he made up 4 steps to prove that a specific microbe causes a specific disease. Okay, so Koch, Haas, agar, so Hess, sorry. They used to use, and they still do, use agar for things like making jelly. So it was actually his wife that got the idea to use seaweed agar. It's a polysaccharide derived from seaweed to grow, use it in the petri dishes to grow bacteria. Okay, and then this guy, Metcalfe, discovered the phagocytosis. Graham, we still use this procedure today. Graham staining, we're going to do it in class. Escher discovered E. coli. Petrie made the petri dish. Kitasoto discovered Clostridium tetani, which we have a Texas tetanus vaccine. My grandmother's sister died of tetanus. So it has not been that long, and I feel like people don't understand how beneficial vaccines have been in our life. Okay, then we have von Behring, diphtheria antitoxin, and then Ehrlich, theories of immunity. Okay, we're going to stop here, and then we'll pick it up in part 2.